CRISPR meets ChatGPT, the AI revolution in gene therapy design. Picture this, the same AI that writes poems and answers trivia is now helping design life-saving gene therapies. That's exactly what's happening as CRISPR, the Nobel-winning gene editing tool, teams up with large language models LLMs like ChatGPT. Together, they're tackling challenges that once seemed insurmountable, bringing us closer to cures for genetic diseases, cancers, and more. CRISPR, pioneered by scientists Jennifer Doudna and Emmanuelle Charpentier, acts like molecular scissors, snipping DNA with remarkable precision. But let's be honest, it's not perfect. Sometimes it cuts the wrong genes, those pesky off-target effects, and designing the right tools for each job can feel like solving a Rubik's Cube blindfolded. Enter AI. By crunching mountains of biological data, tools like ChatGPT are now optimizing CRISPR designs, predicting outcomes, and even dreaming up new gene editing proteins that nature never imagined. The result? Faster, safer, and smarter therapies. Take OpenCRISPR-1, for example, the first gene editor fully designed by AI. It's like having a master engineer in the lab, tweaking CRISPR components to work better than natural systems. Or CRISPR-GPT, a specialized AI assistant that helps researchers design experiments in minutes instead of months. These aren't sci-fi fantasies, they're real tools already accelerating breakthroughs. But let's not gloss over the elephant in the room. Mixing AI and gene editing raises big questions. Who gets access to these high-tech treatments? How do we stop bad actors from misusing the tech? And who's responsible if an AI-designed therapy goes sideways? These aren't just debates for scientists in lab coats, they're conversations we all need to join. The bottom line, we're standing at the edge of a new era. By marrying CRISPR's precision with AI's brainpower, we're not just treating diseases, we're reimagining medicine itself. But to get it right, we'll need collaboration, transparency, and a healthy dose of caution. After all, the future of healthcare isn't just about smarter tech. It's about building a world where these tools serve everyone, ethically and equitably. Historical context. CRISPR and AI might seem like an odd couple, but their partnership makes perfect sense. CRISPR burst onto the scene a decade ago, turning genetic engineering from a slow, pricey process into something faster and cheaper. Meanwhile, AI models like GPT were learning to write like humans. When researchers realized these AIs could also speak the language of DNA, the lightbulb moment happened. What if AI could help us hack biology? Early experiments showed promise. AI began predicting which CRISPR tools would work best, reducing trial and error lab work. By 2023, projects like OpenCRISPR-1 proved AI could invent entirely new gene editors. It's like teaching a computer to cook, except instead of recipes, it's whipping up custom-made medical tools. How it works: CRISPR plus AI, better together. CRISPR's role: think of CRISPR as a GPS-guided scalpel for DNA. It finds specific genes and makes precise edits. But even the best GPs sometimes takes a wrong turn, hence those off-target cuts. AI's superpower: here's where ChatGPT-like models shine. Trained on billions of DNA sequences and research papers, they spot patterns humans miss. Need a guide RNA that only targets the gene causing sickle cell anemia? Ask the AI. It'll sift through options faster than a lab team could blink. The dream team, together, they're like a superhero duo. CRISPR does the heavy lifting, while AI plays strategist, optimizing designs, flagging risks, and even drafting lab protocols. Tools like CRISPR-GPT act as a 24/7 assistant, answering questions like, what's the best way to edit this cancer gene? Real-world wins and lessons learned. OpenCRISPR, this AI-designed editor, isn't just a lab novelty. It's been shared openly, letting researchers worldwide tackle diseases with upgraded tools. CRISPR-GPT in action: scientists using this tool report fewer dead ends in experiments. One team used it to design a therapy for a rare genetic disorder in days, not years. Ethical speed bumps: when an AI suggested a risky edit, it sparked debate. Should machines have a say in human DNA? The takeaway: AI is a tool, not a replacement for human judgment. The road ahead: the future looks wild. Imagine AI designing personalized gene therapies tailored to your DNA, or predicting how edits might affect your health decades later. But let's keep our feet on the ground. For every breakthrough, we need clear rules. Who's accountable if an AI-designed treatment fails? Fair access, preventing a world where only the wealthy get cutting-edge cures. Public trust, demystifying the tech so people aren't left wondering, wait, AI is editing genes now? This isn't just about smarter science, it's about shaping a future where technology heals, unites, and empowers. And that's a story worth writing together. Bonus Q as How are large language models, LLMs, like ChatGPT currently being used to assist in the design and optimization of CRISPR-based gene therapies? Large language models, LLMs, like ChatGPT, are increasingly being utilized to assist in the design and optimization of CRISPR-based gene therapies, leveraging their ability to process and generate complex information, automating CRISPR design processes. LLMs such as CRISPR-GPT have been developed to automate and enhance the design of CRISPR-based gene editing experiments. These models are augmented with domain-specific knowledge and external tools, enabling them to outperform general LLMs in gene editing design tasks. For instance, CRISPR-GPT has been shown to excel in human expert evaluations, demonstrating its ability to streamline the design process and reduce the need for extensive manual intervention. Modeling CRISPR-Cas systems, advances in protein-based LLMs have opened up possibilities for modeling CRISPR-Cas systems without requiring extensive training data. This capability is particularly valuable for identifying and optimizing CRISPR-Cas systems, which are critical for precise gene editing. By leveraging these models, researchers can predict and design novel Cas proteins that may be more versatile or efficient than those found in nature. Enhancing experimental efficiency, the integration of LLMs into CRISPR research helps address the complexity of experimental systems. By providing insights into CRISPR technology and suggesting optimized experimental designs, LLMs can reduce the time and resources required for developing efficient gene editing systems. This is particularly important given the intricate nature of genome engineering and the need for precise modifications. Generating novel gene editing tools, LLMs are also being used to generate new gene editing tools. For example, AI-designed gene editors, inspired by natural CRISPR systems, have been developed using generative AI techniques. These tools could offer greater versatility and functionality, expanding the potential applications of CRISPR technology in biomedical research. Bridging knowledge gaps, while LLMs have shown promise, they often struggle with domain-specific biological knowledge. To address this, models like CRISPR-GPT are specifically tailored to incorporate relevant biological data and tools, enabling them to provide more accurate and actionable insights for CRISPR-based therapies. LLMs like ChatGPT are playing a transformative role in CRISPR research by automating design processes, modeling CRISPR systems, enhancing experimental efficiency, generating novel tools, and bridging knowledge gaps. These advancements are accelerating the development of next-generation gene therapies, though challenges remain in ensuring the accuracy and specificity of these models for biological applications. What are the primary challenges or limitations associated with using large language models like ChatGPT in the design and optimization of CRISPR-based gene therapies? The integration of large language models LLMs like ChatGPT into the design and optimization of CRISPR-based gene therapies presents both transformative opportunities and significant challenges. Accuracy and reasoning limitations, LLMs, including ChatGPT, face challenges in ensuring accuracy and robust reasoning in gene editing experiment design tasks. For instance, while CRISPR-GPT and ChatGPT 3.5/4 have been evaluated for their performance in gene editing tasks across modes like metamode, automode, and QA mode, their scores in accuracy and reasoning highlight room for improvement. This underscores the need for more advanced LLMs tailored to the complexities of biological systems. Biological complexity and contextual understanding, LLMs are designed to process and generate text, but biological sequences and gene editing tasks require a deep understanding of molecular biology and context-specific knowledge. While LLMs can assist in shortening development timelines for gene therapies, their ability to fully grasp the nuances of CRISPR mechanisms, such as off-target effects or immune responses, remains limited. Model size and computational demands. The size of LLMs poses technical challenges, as they require significant computational resources for training and deployment. This can limit their accessibility and scalability in resource-constrained settings, such as smaller research labs or developing countries. Immune responses and delivery challenges. CRISPR-based therapies face inherent biological challenges, such as immune responses to the CRISPR-Cas9 system and difficulties in delivering gene editing components to target cells. While LLMs can aid in designing strategies to mitigate these issues, e.g., targeting immune-privileged organs like the eyes or brain, they cannot fully address the biological complexities involved in gene delivery and immune evasion. Ethical and safety concerns. The use of LLMs in designing gene therapies raises ethical questions, particularly regarding the potential for unintended consequences, such as off-target effects or misuse of the technology. While LLMs can accelerate research, they must be used cautiously to ensure safety and ethical compliance. Integration with experimental validation. LLMs can generate hypotheses and design experiments, but their outputs require rigorous experimental validation. The gap between computational predictions and real-world biological outcomes remains a significant limitation. While LLMs like ChatGPT offer promising tools for advancing CRISPR-based gene therapies, their limitations in accuracy, biological understanding, computational demands, and ethical considerations highlight the need for continued refinement and integration with experimental research. Addressing these challenges will require interdisciplinary collaboration between computational scientists, biologists, and ethicists. Can you explain how large language models like ChatGPT are being used to assist in the design of CRISPR-based gene therapies? What specific tasks or processes do they contribute to? Large language models, LLMs like ChatGPT, are increasingly being utilized to assist in the design of CRISPR-based gene therapies, contributing to several specific tasks and processes. Designing guide RNAs. CRISPR systems rely on RNA molecules, known as guide RNAs (GRNAs), to specify their target sequences within the genome. Madani's team developed an AI model specifically to design these GRNAs, which are critical for the precision of CRISPR-based gene editing. This AI model was trained on diverse CRISPR systems, enabling it to generate millions of new CRISPR protein sequences and guide RNAs, thus expanding the toolkit available for gene editing experiments. Experimental design assistants, LLMs like CRISPR-GPT, have been evaluated for their ability to assist researchers in designing CRISPR experiments. In a study, 12 CRISPR experts tested the model's performance in tasks related to experimental design. The model was assessed on 4 key aspects: accuracy, reasoning, completeness, and conciseness. This demonstrates that LLMs can provide valuable support in structuring and optimizing gene editing experiments, though they still require expert oversight. Target site identification. Machine learning models, including LLMs, have been used to analyze viral genome data to identify high-specificity target sites for CRISPR editing. This is crucial for ensuring that the gene editing process is both effective and safe. LLMs also facilitate the design of therapeutic protocols, guiding researchers on how to implement CRISPR-based therapies with minimal off-target effects. Limitations of general-purpose LLMs. While LLMs show promise, general-purpose models like ChatGPT are not inherently equipped to design biological experiments. They lack the specialized knowledge required for tasks such as gene editing experimental design, which demands a deep understanding of CRISPR technology and the biological systems involved. This highlights the need for domain-specific fine-tuning or hybrid models that combine general-purpose LLMs with specialized biological knowledge. Insights into gene expression. Models like GET, which functions similarly to LLMs, have been used to uncover new insights into gene expression, aiding in the development of targeted therapies. These models learn patterns from vast datasets, much like how ChatGPT learns language structures, and can be adapted to predict how genetic modifications might influence gene expression and cellular behavior. LLMs like ChatGPT are contributing to CRISPR-based gene therapy design by assisting in the creation of guide RNAs, optimizing experimental protocols, identifying target sites, and providing insights into gene expression. However, their utility is currently limited by their lack of specialized biological knowledge, necessitating further development of domain-specific models or hybrid approaches. How might the use of LLMs in designing CRISPR-based gene therapies influence public perception and trust, particularly in light of historical controversies surrounding genetic engineering such as GMOs or designer babies? Are there specific strategies to mitigate potential public distrust? The integration of large language models, LLMs like CRISPR-GPT, into the design of CRISPR-based gene therapies has the potential to significantly influence public perception and trust, particularly given the historical controversies surrounding genetic engineering, such as GMOs and designer babies.